Hello, everyone, and welcome to today's presentation, Accelerate Your Research with Luminex's XMAP INTELLIFLEX, the XMAP technology you trust, plus the versatility you need. I'm Sherry Dunbar, and I'm the Senior Director of Scientific Affairs Programs for Luminex, and I'm very excited to have the privilege of speaking to you today about the XMAP INTELLIFLEX instrument, which is the newest multiplexing platform available from Luminex. First, let me take a moment to introduce you to or reacquaint you with Luminex and review some of the key milestones in the history of our company. The company began in 1995 with a 64-plex of color-coded microspheres and a software to run them on a flow cytometer. But it was in 1999 that we shipped our very own Luminex 100 analyzer, complete with a full 100-plex of bead sets. We called this the 3 5s, which meant that it was 1/5 the size of a flow cytometer. It was 1/5 the price of a flow cytometer, but 5 times faster. So this instrument was specifically designed and optimized for running multiplexed assays using our XMAP microspheres. It was also in 1999 that I joined the company, and so I've been able to experience all of our growth and the advances of our technologies over the years. Luminex works closely with our partners in diagnostics and in research, and it was one of our diagnostic partners who achieved the first IVD-cleared assay on XMAP technology. I believe that this was in 2001, and it was for HLA tissue typing. In 2005, we received IVD clearance for our XTAG cystic fibrosis assay, and this was the first multiplexed molecular test for CF that was cleared for diagnostic use. In 2007, we ventured into the infectious diseases arena and received IVD clearance for the first multiplexed molecular test for respiratory viruses, which was the XTAG respiratory viral panel. The FlexMap 3D instrument came out in 2009, and it was a huge advancement in XMAP technology with the ability to now analyze 500 different colored bead sets at the same time in a single reaction well. We then began to develop real-time PCR applications, and we received clearance for our HSV1 and 2 multiplexed real-time PCR assay using the multi-code iso-based technology from Aerogen. In 2013, the XTAG gastrointestinal pathogen panel became available for in vitro diagnostics and was the first molecular multiplex test for the detection and differentiation of bacterial, viral, and parasitic gastrointestinal pathogens. In 2015, we launched our own sample-to-answer cartridge-based real-time PCR instrument, the Aries, and with it, the Aries HSV1 and 2 assay. Then, with our acquisition of Nanosphere, we gained the ability to do higher plex sample-to-answer molecular testing and could now offer both targeted and syndromic sample-to-answer assays. In 2019, we went back to our roots with the acquisition of the flow cytometry business from Millipore Sigma. And we have several flow cytometers for applications such as cell counting, basic cell viability, and cell health analyses, and also imaging flow cytometers that add quantitative microscopy imaging for every cell that's analyzed. Of course, 2020 marked the start of the COVID-19 pandemic, and we were fortunate to be one of the companies able to provide diagnostic solutions for SARS-CoV-2 detection. We developed EUA-authorized nucleic acid assays for detection of SARS-CoV-2 on all of our platforms, and also a multi-antigen serological assay for measuring virus-specific IgG. Then in 2021, we launched our new XMAP INTELLIFLEX instrument, which is what I'm going to be speaking to you about today. But also in 2021 was another momentous event, which after 26 years in business, Luminex was acquired by DiaSorin. So now we are Luminex, a DiaSorin company. And together with our new parents, we're really looking forward to all that we're going to be able to achieve and what we're going to be able to deliver to laboratories in the years to come. Until the launch of INTELLIFLEX, 2 flow analyzers have really been the workhorses of XMAP technology assays. The Luminex 200 became available in 2007, and it was our first upgrade to that original Luminex 100 analyzer. The Luminex 200 can multiplex up to 100 assays in a well and can read a 96-well plate in 45 minutes. This is 12,800 results per hour. The XMAP 3D came out in 2009 and really advanced the technology for the high-throughput laboratory. It can run a full 500 plex of multiplexed bead assays, and through the dual fluidics pathways, it can process a 96-well plate in about 20 minutes. It can also run 384-well plates, and it can read a whole 384-well plate in about 75 minutes. So with XMAP 3D, you can get 144,000 results per hour in 96 wells, or 153,600 tests per hour in the 384-well format. Now IntelliFlex. IntelliFlex builds on the best of the Luminex 200 and the best of XMAP 3D, but in a modern, compact instrument. IntelliFlex has a very intuitive software that has flexibility and allows customization for your specific workflows and even your preferred file formats. File formats are available that are compatible with our partners' analytics software, as well as other common data analysis packages. All of this is intended to provide an overall better user experience. It's still delivering the reliable, sensitive, and high-quality data that people have come to know and expect from XMAP technology. But now with a new second reporter channel, you can simultaneously measure 2 different parameters for each of your analytes. So therefore, you can essentially double the amount of data that you get from the same number of wells. IntelliFlex is based on 3 main concepts: the high quality data, and note that the system is completely compatible with the 1,000+ assays that are already available for XMAP technology from Luminex and from all of our partners. The new dual reporter capability, which I'm going to share some data about and examples about later in the presentation, and the flexibility to customize the look and feel of the user interface and the format of the data output. Before we dive into the data, though, I'd like to spend a few minutes reviewing key concepts of XMAP technology that we can build on as we learn about IntelliFlex. Let's start with some fast facts. After 27 years, XMAP is certainly a proven technology, and we really are the world leader when it comes to bead-based multiplexing. At last count, there are more than 58,000 peer-reviewed publications that use XMAP technology. And these references use XMAP technology products from Luminex, but also from all of our partners and all of their XMAP products. Over 20,000 instruments have been placed so far in laboratories around the world. And at last count, more than 1,300 RUO assay kits have been developed, and more than 100 XMAP-based assays have received 510(k) clearances for diagnostic use. But these stats increase almost daily, so I'm sure that this slide is probably already out of date. So let's talk about some of the ways XMAP technology is used. Virtually any binding event can be applied to the microsphere surface. If there is a way to capture an analyte and a way to detect it, you can run it on XMAP technology. And this same technology works great for both proteomic and genomic applications. Common assay formats include the capture sandwich immunoassay, where a specific capture antibody, usually a monoclonal, is covalently coupled to the bead surface and can bind to the analyte in the sample. Then a labeled detection antibody can be used to quantify the binding events that have occurred on the microsphere surface, forming an analyte sandwich. The competitive immunoassay, shown in the middle here, is an assay format that we typically use for small analytes and if only one antibody is available. We can do this in 2 ways on the microspheres. As shown here, we can use an antibody-coupled microsphere and a labeled analyte to compete with that analyte in the sample for binding to the microsphere. The signal decreases as the amount of the unlabeled analyte in the sample increases. Another way to do this is to couple the analyte to the bead and then use that to compete with the analyte in the sample for binding to a labeled detection antibody. The indirect assay format shown on the right is used for serological tests where a specific antigen is coupled to the bead and then can be used to bind to specific antibodies present in the sample. These captured antibodies can then be detected and quantified using a reporter-labeled anti-immunoglobulin detection antibody. XMAP multiplexing has been used for numerous applications in proteomics, including identification of secreted proteins, determining the mechanisms of action in metabolic and enzymatic pathways, drug monitoring, looking at protein-protein interactions, and many others. On the nucleic acid side, shown in the bottom half, we couple a capture probe onto the bead, and then we use a variety of assay chemistries for target detection. One of the most common assay formats is direct hybridization of a labeled PCR product. The sequence-specific capture probe is covalently coupled to the bead and used to capture the target that has been amplified and labeled from the original sample. We also have our Magplex tag microspheres, which are pre-coupled with unique anti-tag capture sequences. The assay chemistry, whether it's PCR, primer extension, or oligonucleotide ligation, serves to incorporate the complementary tag sequence and a label for detection into the nucleic acid target if it was present in the original sample. The hybridization is so specific that it can discriminate to the single nucleotide level. So it performs very well for SNP genotyping and even microRNA analysis. And with the appropriate standards and controls, it can also be quantitative or semi-quantitative. And it works really well for applications such as gene expression analysis and measuring copy number variation. Here's an example of how XMAP multiplexing compares to other common platforms. And we'll use the analysis of 21 cytokines in 80 samples as our example. As compared to a singleplex technology such as ELISA, XMAP allows you to run all of your assays simultaneously in solution with a small reaction volume. So you will use less sample, and it will take less time and less work to generate results. You will have a lower cost for the reagents and the consumables, plus you'll generate more data, and really better data that I'm going to explain to you further in a moment. If we're comparing to planar arrays, you're going to have a faster binding kinetics because the beads are in suspension and with a very high surface to volume ratio that can provide better sensitivity. So to analyze 21 cytokines from 80 samples, you would need 42 96-well plates if you were using a singleplex ELISA. If you were using a planar array system, you would need probably at least 3 96-well plates if you could have up to 10 spots per well. But with XMAP technology, you only need 1. In the publications that are cited here, you can read about some studies where these investigators compared the performance and the workflows for different methods for cytokine analysis. And there are links to these papers as well on the Luminex website. So let's talk about Luminex's partners. Our partners have really taken the technology to the next level and beyond. As I mentioned at last count, there were over 1,300 assays available, as well as custom assay capabilities and various analysis software packages from different Luminex partners. Our Kit Finder app, shown on the left, is available on the Luminex website, and it can help you find an already available assay kit for your analytes and for your species of interest. Some examples from our partners include products from Biorad, Biotechne, Millipore Sigma, and Thermo Fisher. And these include multiplex assay kits in both 96 and 384 well formats, as well as custom assay services and software packages for analyzing the data. So here I'd like to encourage you to check out all of their websites to see all that's available for XMAP technology. Now let's review how the detection works on our flow analyzers. The completed assay that contains the multiplex beads is aspirated into the sample cuvette in a rapidly flowing fluid stream. Hydrodynamic focusing confines the beads to the center of the sheath fluid core and sends the beads past the lasers one at a time. Each bead is excited with the red laser to measure the unique combination of dyes inside the bead, which identifies the bead region, and hence the specific assay that's being measured on that bead. The green laser excites and measures the surface orange fluorescence to quantify the amount of analyte that has been bound to the microsphere surface. So the instrument software calculates the reporter signal on the microsphere surface. And the reporter dye that's present is correlated to the amount of analyte that was present in the sample. The reporter dye, which we call RP1, excites at 532 nanometers and it measures the emission at 565 to 585 nanometers. 50 microspheres, at least, of each region are individually interrogated. The median fluorescent intensity is calculated from all of the events, and that's what's reported by the software. And by taking the median, the effect of bead carryover is eliminated because outliers are excluded from the analysis as depicted at this chart on the bottom right. So just to recap, for each reaction, we typically measure a minimum of 50 beads per set of each bead color or bead set that is in the reaction. We then determine the median of this fluorescent intensity that was measured for those beads. We call this value MFI. And unlike an ELISA where you're measuring the sum of the signal in the well, each bead is measured as a replicate of the reaction. So for each well, we are essentially measuring 50 replicates per assay and then reporting the median of those measurements. So the data are very robust and the median ensures data integrity. The median really is the best statistic to use because unlike the mean, it cannot be skewed by a few events that may have been carried over from a previous well. Okay, so now that we've been through all of these basics of Luminex and XMAP technology, I think we're ready to dive into the IntelliFlex in more detail. We have two models for IntelliFlex. The standard model, which has much of the same features that you're used to on FlexMap 3D, such as the 500-plex capability, a single green reporter laser. It's compatible with both 96-well and 384-well plates, but it also has an additional decade of dynamic range. So now there's 5.5 logs of dynamic range in the Reporter 1 channel. And with an integrated PC and a touch screen, it has a really compact footprint, as well as hardware features that make it really easy to load and unload a plate. It's designed to be modular with components that are easy to access for our field service engineers when they come to do your preventative maintenance. The second option, the DRSE model, has all of this, plus a second reporter channel that employs a violet laser. This new reporter channel has about 4.5 logs of dynamic range, and this model also has a side eject plate carrier that makes it really easy to connect to laboratory automation systems. Now let me describe the optics in IntelliFlex. In the single reporter system shown on the left, we have our avalanche photodiodes, or APDs, and our doublet discriminator, DD, for bead classification through the red laser. The 532 nanometer green laser excites and sends the reporter fluorescence to the photomultiplier tube, or PMT, to measure reporter 1 emission at about 565 to 585 nanometers. And it will also send some reporter signal to the extended dynamic range, or EDR detector, so that the reporter signal can still be quantified even if the PMT has reached saturation. In the dual reporter model shown on the right, we have an additional 405 nanometer violet laser that excites the second reporter dye and measures emission at 421 to 441 nanometers in the second photomultiplier tube. Okay, as an example of IntelliFlex performance, I have some data that was kindly provided by Thermo Fisher. This chart shows the results for IL-8 from their 45-plex pro-Cartiplex cytokine panel. You can see here on the left that when the standard curves generated on IntelliFlex were compared to the Luminex 200 and the FlexMap 3D instruments at all of the possible calibration settings or operating modes, the results were virtually identical. The standard curves are laying right on top of each other. And if you look on the right at the data for a variety of different samples, the results were also very similar to that on the classic XMAP flow instruments. This will be important for those of you who are already using an XMAP instrument and assays, because you have all of the settings that will provide you the option of seeing the results and the MFI values that you're used to seeing But as well, you can try some of the new data acquisition modes that are only available on IntelliFlex. So now let's talk about some applications for IntelliFlex. Here's a cartoon of a serological application that we would use to measure antibody response. This has been particularly useful now in the time of COVID, as we've been looking at antibody responses to vaccines as well as natural infection. So just to remind you that in a single reporter instrument, if you were interested in looking at different antibody isotypes to a specific antigen, you would have to run 2 separate wells. In this example, let's say we want to measure IgG and IgA that is specific for the green hexagon antigen. So all of the antibody isotypes that are present in the sample are going to bind to the antigen on that bead, but we could only detect one at a time. So in well 1, we can use a phycoerythrin or PE labeled anti-IgG detection antibody, and the red laser would classify the bead set in which antigen is being tested. The green laser measures that IgG that is bound to the surface and is being detected with that PE labeled anti-immunoglobulin antibody. Okay, but what about IgA? So now you have to do a separate well. So you set up the identical reaction in the second well, but in this well, we're going to use a PE labeled anti-IgA detection antibody to detect how much of the antigen specific IgA was bound. So this is how we would typically do this. Okay, but now with IntelliFlex, it's a whole new ball game because of the dual reporter option. The dual reporter gives you the ability to measure 2 antigen-specific antibody isotypes in a sample from only one well. As before, the antigen is coupled to the microsphere, and then the microspheres are allowed to incubate with the sample. So all the antibodies in the sample that recognize and can bind to that antigen are captured onto that antigen-coupled bead. So whatever isotype it is. But in this example, we can add a PE labeled anti-IgG detection antibody and a brilliant violet labeled anti-IgA detection antibody at the same time. And this allows measurement of both the antigen-specific IgG and antigen-specific IgA that's bound to the bead in the same well at the same time. So here's some example data for SARS-CoV-2 antibody responses where we used our own XMAP SARS-CoV-2 multi-antigen IgG kit, but we substituted different detection antibodies and different reporters to be able to measure IgG and IgM at the same time in our IntelliFlex dual reporter instrument. 3 runs were performed in parallel using the dual reporter setting on serial dilutions of positive serum. In panel A, you see the IgG-PE signal when it was the only detection antibody present in the reaction, and the background levels in the reporter 2 or RP2 channel. In B, you see the IgM super bright 436 signal when it was the only detection antibody present, with low, less than 100 background level MFI in the reporter 1 or RP1 channel. And then in C, you have the responses when both detection antibodies were present in the at the same time in the same reaction, which is shown in the solid lines. And then as shown by overlaying this single reporter data from A and B in the dotted lines, you can see that the results are nearly identical. So this really showed that you can use the 2 different reporter dyes at the same time in the same reaction without any kind of interference between the 2 channels. Next, we have data on serum samples that were drawn at various days post-SARS-CoV-2 RNA vaccination. On the left, we have dual reporter results for IgG-PE and IgA Brilliant Violet 421 for 9 replicate reactions, looking at the vaccine response to the receptor binding domain of the viral spike 1 protein. If you compare the signal at day 0 and day 24 post-SARS-CoV-2 vaccination, shown by this black arrow, you can clearly see the kinetics of antibody response in PCR negative COVID-19 naive individuals. In the center graph, we compared the IgG response against different SARS-CoV-2 antigens. Here we're showing the receptor binding domain, or RBD, of the spike protein and the nucleocapsid, or N, of the virus. And here you can easily differentiate individuals who did not have COVID in the blue, where we have background signals on both antigens. And you can distinguish these from individuals who were vaccinated with a spike-specific RNA vaccine in black, where there's a high signal for RBD and no signal for N. And then you can separate these as well from previously infected individuals, which are shown in red, where we have high signal for both RBD and N. Only infected individuals who will have been exposed to N, whereas those vaccinated with a spike vaccine will not. On the right, we have antibody responses over time, which were measured in the single reporter mode for antigen-specific IgA using all the classic XMAP instruments, MagPix, Luminex 200, and FlexMap 3D. And this was compared to the dual reporter mode on IntelliFlex. I've not discussed our MagPix instrument with you today, but it uses magnetic bead capture and imaging to measure the assays, as opposed to the flow-based analysis that we use with the other instruments and we're talking about today. So as shown here, we saw a near perfect correlation between the dual reporter readout using an anti-IgA brilliant violet detection antibody compared to an anti-IgA PE labeled antibody on the other XMAP instruments. And similar results were also seen for specific IgG, with all of the R-squared values at 0.98 for all instances. Here's some published work that came from the University of Rochester that was done using a 3-plex serological assay, also for SARS-CoV-2. But also ACE2 was added as a competitor to serve as a surrogate marker for potential neutralizing antibody activity to SARS-CoV-2. On the left, we have the antibody response MFIs, and on the right, we're looking at the inhibition of antibody binding when ACE2 was added in a competitive assay format. The blue shading shows the reporter 2 responses measuring IgG, and the orange shading shows the reporter 1 IgM response. The green on the right panel is showing the reactions that demonstrated a 30% or greater binding inhibition with ACE2. And so we can see the increase in antibody signals over time from 3 days of symptom onset shown in panel A versus 12 days shown in panel C and 30 days shown in panel E. And on the right, we see ACE2% residual signals at 3 days from symptom onset in panel B, 12 days in D, and 30 days in F. This study and the protocol for this assay was published in the Journal of Visualized Experiments. The citation is down at the bottom of the slide. So you can actually see these results in much more detail from the paper cited below the tables. And the links to this paper are also available through the Luminex website. So this is the first peer-reviewed publication that was done using the IntelliFlex instrument and the dual reporter mode. Another publication from Rush University shows the results of a dual reporter assay measuring antibody isotypes, also for COVID-19. But here they're determining the specificity for the 2 reporter channels. After optimization of primary standard and sample concentrations, they optimized the secondary antibody concentration and the incubation times. With this fully optimized assay, the authors assessed the specificity for the dual channel assays. Assay results of the dual channel assay format with one of each reporter combination designated as the blank was used to illustrate the lack of cross-channel fluorescence or interference, which is what's shown in the charts here. The overall interference was less than 6.5%, but specific interference depended on which antigen and which antibody isotype was being measured. And thus the authors recommend that one should determine the best combinations to use for dual reporter assays. In this case, they suggest measuring IgM with reporter 1, phycoerythrin, and then either IgG or IgA in the reporter 2 channel with one of the violet dyes. This assay and the method was also published, and you can see this in more detail in the paper cited at the bottom or through the link to this same paper that's available on the Luminex website. In this figure from the same study, they measured seroconversion following COVID-19 vaccination. Plots illustrating the relative titers to the spike antigen during the course of vaccination are shown here, with IgG shown in the top in panel A, IgM on the bottom left in B, and IgA in C on the bottom right. The time measured was from pre-vaccination to up to 4 months post-completion. And the time of the vaccination is shown in days relative to completion of the vaccination series. And the general time points of the 2 vaccine dose administration are indicated in the vertical dotted lines. The experiments were performed in the dual channel mode and are described in much more detail in the published protocol. But you're able to see that for multiple patients, and this is just showing 4 of them, you can definitely see the antibody responses with all of the different isotypes that they're measuring. So now I've shown you several examples of dual reporter immunoassays, but you're probably wondering, well, what about nucleic acids? Because the same XMAP technology works great for both. Well, there's nothing published in the peer-reviewed literature yet, but I do have some data to share with you that we published in our XMAP assay development cookbook. On the left, we had a 2-plex assay for 2 different oligonucleotide targets using our magplex tag microspheres. Each target was directly labeled with a different reporter dye, either Alexa 532 for the reporter 1 side or Alexa 405 for the reporter 2 channel. And what you can see here is when we have both targets and both reporters present in the same reaction at the same time, the signal, as shown by the solid lines, is equivalent to what we see when we only had 1 target and 1 reporter present, as shown by the dotted lines. On the right side, we used Alexa 532 for 1 target for the reporter 1 channel, and we used a biotinylated target with streptavidin superbright 436 for the second target for the reporter 2 channel. And we have essentially the same results whether we had 1 or both reporters present, as is shown by the dotted and solid lines respectively. And as you can see, I want to point out Alexa 532 is a brighter dye than Alexa 405, and we're reaching about a 700 MFI versus only 150 MFI. But on the right, you see that super bright is much brighter at about 8,000 MFI versus the 700 of the Alexa 532. So which combination of reporters that you want to use really depends on the assay. So if I had 1 target that was typically in a pretty high concentration, I would probably use the dimmer reporter dye for that one. And then if I have a target that is usually low and very low in concentration, I would try to use the brighter reporter for that one. Note that for dual reporter assays, at least one of the detection reagents must be directly conjugated to a reporter dye, but then the other one can use a biotin streptavidin reporter system. Okay, so here's some more nucleic acid data, and I'm showing you this because while we looked at 2 targets in a 2-plex in the same well and showed no interference, we wanted to have a model system that would look at the performance when we have both reporters actually on the same molecule captured onto the same bead. And so this would allow you to look at 2 similar targets at the same time in a nucleic acid assay, say a pre-messenger RNA versus a spliced mature messenger RNA. Or like on the protein side, a post-translational modification. So in this example, you could have a capture probe targeted to the same shared sequence that would capture both forms, and then a detection reagent like labeled complementary oligos that would be specific for an intron and a splice junction that would be able to differentiate the relative amounts of the 2 species captured onto the same bead. So as a surrogate for that type of measurement, we developed an assay using a DNA target that has been dual labeled with 2 reporter dyes. One is on the 5' end and the other is on the 3' end. On the left, we used a 5' Alexa 532 for RP1, shown in orange, with a 3' biotin and streptavidin superbright for RP2, shown in blue. And again, we see no impact on the performance in the dual reporter system shown with the solid lines as compared to the single reporter measurements in the dotted lines. So now we're showing that, we have 2 reporters, the 2 different reporters, very close together on the same molecule, and there's no interference and the performance is the same. On the right, we saw similar results when we put the RP2 reporter on the 5' end, shown in blue, and the RP1 reporter on the 3' end, shown in pink. So based on these experiments, we find that the dual reporter system will work as well for nucleic acid assays as it does with immunoassays, and it should also work great for dual labels on the same captured analyte target. And with that, I've come to the end of my talk. And so I'd like to thank you for your attention today. And if you have any questions, please put them into the chat and I'll be happy to answer or get you the answer if I don't know myself. Thank you.
Thank you, Sherry, for your informative presentation. We will now start the Q&A portion of the webinar. Our first question from our audience is, what is the highest plex assay achieved for XMAP technology?
Well, I happen to know that all 500 bead sets have been used in assays, at least on the nucleic acid side. There's a bunch of research that comes from the Broad Institute, and it's been published, and they're using all 500 bead sets to do gene expression profiling and looking at combinatorial chemistries for drug work. Probably other applications as well, but that's the one that's like right on the top of my mind. And on the protein side, I know that at least about a 400 plex is being used by multiple companies and institutions in Europe, particularly when you're doing something that's a serological application where you only have one detection antibody, say an anti-IG specific antibody. You could totally do up to 500 plex with that type of a combination. And so I know that the KTH in Stockholm, Sweden is doing really high plex assays, and the Oncommune in Germany is also doing at least 400 plex assays.
Wonderful. Thank you so much. The next question from our audience is, what are some types of assays that can be used with the dual reporter?
Okay, as I was mentioning, I think this is going to be really great for looking at post-translational modifications, such as methylation or phosphorylation. So having the dual reporter system would allow you to measure the total amount of an analyte that's present, but with specific detection reagents, you could also measure the specific sections that have been modified. And so we think that this is going to be a really great application for the dual reporter system.
Wonderful. Thank you so much for your answer. It looks like we have time for one more question. That is, are there resources available to help with assay development?
Oh, that's really a great question. Well, I'm sure you don't want to spend time going through 58,000 publications, but we do have resources. And we have our XMAP Assay Development Cookbook. We just published the 5th edition in January, and it also includes some of these dual reporter examples that I've shared with you today. In addition, our Luminex Explore Lab is a custom assay development laboratory that's available at Luminex to help people who are developing assays, from soup to nuts. Really, you can start at the very beginning, just trying to get help with coming up with your concept, selecting your reagents, coupling, QC, all the way from beginning to end. They're available to help with that assay development work.
Fantastic. Thank you again, Sherry, for your time today and your important research. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. I'd like to also thank our sponsors, Luminex, a DiaSorin company, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.